Good evening, everyone. Today, we would like to present about customized 3D printed stackable cell culture insert tailored with bioactive membranes. Before that, I would like to introduce myself first. My name is NORFAGHIRA KAMAL and my matrix number is 205275.
Hello everyone, my name is Dylan Lauki. My metric number is 206480.
Hello everyone, my name is IMAN ATHIRAH bin MUTALIB and my matrix number 880.
Hi everyone, my name is RASHEEDAH DURRANI binti Muhammad Nazari. My metric number is 207181.
Hello, my name is AMIRAH FARZANA binti ISMAIL and my metric number is 208194.
Thank you, everyone. So I will present about introduction first. Organ system have been employed to model pharmacokinetic and dynamic, first-pass metabolism, and utilities qualities. It was dominated by microfluidic chips, which transfer type of insert for in vitro been explored. Transfer hanging insert used to recreate single barrier, and it also used to interconnect tissue by co-culturing. The advantage of hydrogel is often transparent, which is not always the case for filters. Next, I will present about result. First result is physical limit of stacker insert. They focus on 2 layers stacked to investigate how small stackable will function. Then they aim to minimize the design dimension. Next result is functionalization of the inserts with filter membranes. Hydrophilic PTFE membrane used to modify the bottom of the insert with a microporous filter membrane. Then PTFE membrane was placed on the bottom and secured with a ring. Then I will present about functionalization of the insert with hydrogel membrane. First, the transport characteristic of hydrogel 10%, 15%, 20% were compared with hydrophilic PTFE. The viscosity was controlled by keeping gelatin at 37 degrees Celsius. The gelatin membrane was reversibly physically cross-linked by reducing the temperature to 4 degrees Celsius. All gelatin concentration had similar mechanical properties, thickness, and transport properties. Second method for measuring the thickness, 1 microgram per milliliter, fluorescent sodium salt was added to the gelatin solution as a reference. Next, I will pass to Dylan.
Okay, thank you, NORFAGHIRA. Moving on to the fourth result, which is the biological validation of the dipping approach using transport studies. Okay, so the objective of this study is to test dip casting process for sustaining long-term cell growth using 96 well inserts where CaCO2 cells were cultured and differentiated on different concentration of their thin membranes, namely 10%, 15%, and 20% for 3 weeks to create intestinal barrier models. Okay, so from this, we can observe that F-actin was expressed at the cell-cell border, indicating tight concentration of cells, and the cells showed no apparent morphological differences in different membrane conditions. So since permeability rates, PC, correlate with barrier functions of the cells, Lucifer-Yalow transport method was performed to assess the barrier functions of the cell. And the result shows that LY standard curve estimated average transport is 4.1 plus minus 2.5 nanometer per second in 10%, and 1.5 plus minus 3.4 nanometer per second in 15%, and 0.5 plus minus 1.0 nanometer per second in 20% in week 3. For your information, PC less than 12 nanometer per second describes a well-established intestinal barrier function of CaCO2 cells. So next, for number 5. The biocompatibility of the inserts for mouse intestinal organoid culture was tested by culturing the organoids in 3D matrigel dome cultures on well plate as a control, hanging insert plates with filter, PTFE, and gelatin membranes. On incubation day 1, well plate showed smaller organoids and many smaller clusters. Both PTFE and gelatin membrane showed larger aggregates with a very few smaller clusters. However, gelatin membrane also showed organoids are surrounded by single cells. On day 3, only PTFE showed organoids getting more compact. On day 7, organoids condensed with gelatin membrane being the most condensed, followed by filter membrane and control. And differentiation and maturation were also observed in all the conditions. On day 10, live and dead staining was performed and filter insert is observed to be the most suitable microenvironment with only a few dead cells found in the buds. Next, for the discussion. Here we found out that deep casting procedure is simple, quick, and low-cost technique. And it also produces very consistent properties of gelatin membranes in terms of transport and thickness and support for intestine barriers. For 3D printers, which is either SLA or DLP, it is preferred due to its high resolution and can also be cost-efficient and accessible. And most importantly, it gives freedom to scientists to design their own culture way. However, the downside of 3D printer is its high aspect ratio combined with relatively thin walls of the inserts, and also the capillary effects between the walls and bubble capture. Now, I'll pass on to the next presenter for the method.
So for the method, the first one is fabrication of 3D printed insert. So you have to design 3D printed insert with the different formats and dimension using Fusion 360. And you have to export as STL format file, and you have to process the file. And then you have to print insert in dental LTV1 resin. And then you have to wash the insert in the IPA tank. And then you have to clean twice all the print in fresh IPA for 1 hour. And then you have to air dry the insert, and then you have to post cure in the foam cure UV oven for 2 hours. After that, you have to submerge insert in the sterile water with 30 ml resin to 1 liter water. And lastly, you have to perform a further 15 UV expose for each side for sterilization. The second one is mass transport through membranes. Firstly, you have to test the permeability of 96 well strip insert, and then you have to place insert in the 96 well plate, and then you have to add PBS, and then add ferrocene sodium salt, and you have to perform transport study with 100 round per minute. And then after the incubation, you have to transfer the samples and analyze relative fluorescent unit, which is RFU, and then you have to collect RFU data and analyze using GraphPad Prism. The third one is assembling synthetic hydrogel membrane. The first one, you have to assemble PTFE membrane, and then you have to apply dental LT to the insert walls, and then you have to fix the membrane toward the walls by using printed cylindrical rings. And then you have to mix the gelatin with PBS, and then adjust the solution using NaOH, and then you have to add chloroform to sterilize the solution. And then you have to mix all the components and place the tube in water bath until the gelatin was fully dissolved. Then you have to pour the gelatin to preheated sterile petri dish, and you have to immerse a sterile 3D printed insert in the gelatin solution for 5 seconds, and then you have to press the insert to form a thin hydrogel membrane. After that, you have to place the membrane inside the cells culture plate and place at 4 degrees for at least 20 minutes. After the reversible cross-linking of the gelatin membrane, you have to add MTG solution to the wells with the amount to cover insert well surface. You have to place the plate in the incubator, and then you have to wash the sterilized hydrogen membrane in PBS for 10 minutes, 3 times. And all the insert with gelatin membrane either used for culture directly or stored in the fridge for maximum 1 month. The fourth one is mechanical testing of hydrogen membrane. The first step, you have to prepare gelatin hydrogel disc using the sandwich casting method, and you have to coat 2 ppma slide, and you have to glue 2 1 millimeter thick ppma slide with 2 pairs of 3 millimeter thick PMMA slide, and you have to cast it, and then you have to cross-link the gelatin disc with 1 mm thickness in the fridge for 4 degree. And after that, you have to carefully remove the gelatin disc by fusing the MTG solution between the base slide, and you have to incubate. And then you have to wash the gelatin disc with PBS twice and store in closed petri dish with PBS. And you have to load manufacture sample, and then you have to perform frequency sweep test at the strain of 0.01%, and then you have to perform strength sweep test from 0.2 to 200%, and use the result to evaluate the material properties. And I will pass to DURRANI.
Thank you. Then I will continue with the method thickness measurement of hydrogel membrane. So firstly, fluorescent gelatin was prepared by mixing 15% gelatin powder in 1 microgram per ml fluorescent sodium salt. And then 6 liquid coupled slides were created by using the similar sandwich method described to form the standard curve of fluorescence intensity of different heights provided by stacking cover slips onto a 1 mm thick glass microscope slide. Then 500 microliter fluorescence gelatin was pipetted onto the base slide and covered with the other pair of the base slide. And then inserts 96 and 48 well format strips were fabricated as described and placed onto a base slide. And then gelatin membrane were imaged with Zeiss Axial Observer Z1 epifluorescence microscope at an emission wavelength range of 495 nanometer to 570 nanometer by a LED laser with filter set 38 HE and within 150 until 900 micrometer thickness range. And lastly, all the files were processed with Zeiss Zen Blue 3.4 light digital imaging software to correlate membrane heights. And then the method swelling ratio of hydrogen membranes. 15% gelatin membranes were assembled to 48 well format gelatin inserts. Membranes were punched with 3 mm diameter biopsy skin punch as freshly cross-linked and swollen in PBS for 1 hour at 57 degrees Celsius and dehydrated overnight at room temperature. And then lastly, the thickness of punch membrane disc was measured by using micrometer screw and the swelling ratio was calculated. For the next method, cell types and culture conditions. Human epithelial colon carcinoma cells were cultured in T75 cell culture flask in high glucose dulbicose DMEM medium. And then the KHIRUL cells were seeded on hydrogen membranes at a density of 80,000 cells per cm squared and incubated for 3 weeks. Human umbilical vein endothelial cells were cultured in endothelial culture growth medium with 10% FBS and 1% PS. And then the HUVEC cell cultures were seeded on gelatin micromembranes and material-coated PTFE membranes at a density of 100,000 cells per cm squared and incubated for up to 1 week. And then cells were sprayed with trypsin, ethylene diamine tetraacetic acid, EDTA, for 3 to 5 minutes upon 90% confluency. And then mouse intestinal organoids were cultured, and then intestinal crypts were thawed, centrifugated and suspended in metro gel and transferred into 24 well PTFE membrane assembled insert plates. Intesticles, mouse organoid growth medium supplemented with 1% PS was overlaid on the gel in each well after polymerization. And then lastly, all cell lines and organoids were maintained in an incubator with the culture medium replaced every 2 days. And last methods, actin, nuclei, live or dead stains. Firstly, samples were washed for 15 minutes in PBS, followed by fixation in 2% PFA in PBS for 2 minutes and fixation in 4% PFA in PBS for 30 minutes at room temperature for F-actin and Cook's 3342 staining. And then PFA was aspirated from the samples and washed 3 times in PBS. Samples were incubated with the stain for 20 minutes at room temperature. Next, the samples were washed 3 times in PBS and left in PBS. Samples were washed 3 times in PBS and stained in 500 microliter of a solution with ethidium homodimer 1 and calcium AM for 1 hour at room temperature for live or dead staining. And lastly, the samples were washed in PBS 2 times and then kept in PBS to keep the hydrogel growth matrices moisturized. And then I will pass to AMIRAH.
Okay, thank you, Durrani. So first we'll start with microscopy imaging. So there are only 4 steps, which is first we will use a Zeiss Primovet microscope to obtain a phase contrast bright field micrograph, which the objective lens suggested is plain achromite 4 times. And then second, use Zeiss Axial Observer Z1 epifluorescence microscope to stain image. So at the side you can see the suggested objective lens also. Then thirdly, feed the image with a scale bar and process them in Zen Blue 3.4 lit digital imaging software and image. And lastly, we will perform surface area analysis on organoids by using the macro. So next is the statistical analysis. First, we will present the data as a sample sizes and means and standard deviation SDS. Second, we will be using micro Excel to calculate and analyze it using a graphite prism. For anomaly testing, we will use Sephirovic. And third, analyze the data by using an ordinary one-way ANOVA or two-way ANOVA with alpha 0.05 with post-hoc to case multiply comparison test for equality. Fourth, obtain p-values using Barnett's correction and determine significant difference when p-value is less than 0.05. And lastly, we will perform F-test for well-t-test to compare variances of membrane thicknesses and highlight the significant differences with symbol or figures. And lastly, transport of lucifer yellow. First, we will place the single insert in a 96 well plate and add a 235 microliter of preheat HBSS transport buffer, sodium bicarbonate, HEPES, and BSA to the basolateral site, and then add a certified microliter of 60 micromolar lucifer yellow to the apical site in each insert. Secondly, perform the transport study at 97 degrees with 100 rpm shaking under a dark condition for 2 hours. And thirdly, transfer the 100 microliter sample from the first insert, 50 microliter sample from the second insert, and 50 microliter HBSS transport buffer to a 96 well plate. For analyze the relative fluorescence unit or RFU emission at an excitation wavelength of 438 nanometer and measuring emission at 536 nanometer. And lastly, we will calculate the permeability coefficient, PC, which can be seen the equation beside it, which VR is the receiver volume, A is the membrane growth area, CI is the initial apical concentration, CF is the final receiver concentration, and T is the assay time in seconds. So I think that's the last one of the method and the end of our presentation. So that's all from us. Thank you.